Over the past 2 decades, the global pharmaceutical landscape has shifted. As patents for originator biological medicines expire, a surge of highly similar versions, known as biosimilar products, is entering the market at an accelerating pace. It is easy to assume these are simply generic biologics, and that they can be monitored using the exact same post-market safety protocols as small molecule generics. That is a dangerous systemic error. A generic drug is a chemically synthesized small molecule. It is an exact, identical copy of its branded counterpart. Biologics are completely different. They are large, complex proteins derived from living cells. Because of this intricate manufacturing process, it is physically impossible to create an exact replica of a biologic. A biosimilar is approved by demonstrating it has no clinically meaningful differences from the reference product, but slight variations always exist. Standard pharmacovigilance for generic drugs relies heavily on the assumption of identical chemical replication. If the molecule is mathematically identical, the safety profile remains the same. Because identical replication of biologics is impossible, the safety net that applies to generic drugs does not exist here. The post-market surveillance architecture for biosimilars has to be entirely re-engineered to monitor physical variants. In the manufacturing of biological medicines, there is a foundational axiom: the process is the product. The cell lines, culture conditions, and purification processes used by biosimilar manufacturers will differ from the originator. These variables are highly sensitive. Advanced analytical methods have limits. Even rigorous scanning cannot detect every clinically relevant structural difference between a biosimilar and its reference product. These minor undetected changes in folding or post-translational modifications introduce latent safety variations. Often, these variations only become apparent through long-term clinical use across large human populations. The primary, unique risk created by this variability is immunogenicity, the potential for a therapeutic protein to elicit an unwanted immune response in a patient. Ultimately, the human immune system is the final, most sensitive analytical tool for detecting batch-to-batch variants in biological manufacturing. We have definitive historical proof of this manufacturing sensitivity. In the late 1990s, Johnson & Johnson manufactured an originator biologic called Eprex, used to treat anemia in patients with chronic kidney disease. The manufacturer made minor formulation changes. They replaced human serum albumin with polysorbate 80 as a stabilizer and changed the syringe stopper material. The new Eprex formulation triggered rapid generation of neutralizing anti-drug antibodies. These antibodies aggressively attacked the drug and cross-reacted with patients' endogenous erythropoietin. Consequently, red blood cell production dropped to zero, causing a spike in life-threatening pure red cell aplasia. This severe clinical failure forced regulatory agencies worldwide to permanently strengthen pharmacovigilance requirements for biological products. These unique risks led directly to specific regulatory pathways, like the Biologics Price Competition and Innovation Act, signed into U.S. law in 2009. Today, modern regulatory agencies like the FDA require stringent, dedicated frameworks specifically designed to catch these complex immunogenic events. The historical Eprex anomaly definitively proves that a single microscopic variance in formulation can trigger a sudden, catastrophic autoimmune reaction in a patient. Pre-market clinical trials, short and limited in scope, are insufficient on their own to guarantee safety. This need for rigorous tracking is mathematically amplified by the regulatory concept of extrapolation. Agencies may approve a biosimilar for multiple reference indications based entirely on clinical trial data generated for only one specific indication. While justified by molecular comparability, this creates an immediate data gap. Real-world safety data for extrapolated indications literally does not exist yet. This leaves regulators temporarily blind to rare, indication-specific safety signals. To actively fill this void, agencies like the European Medicines Agency require mandatory risk management plans and post-authorization safety studies immediately upon launch. Because of extrapolation, post-market surveillance for a biosimilar is not a passive secondary safety net. It operates as the primary clinical validation mechanism for the majority of its approved uses. Tracking these rare signals requires precision, which brings us to the most critical logistical bottleneck in biosimilar safety: the traceability crisis. Standard spontaneous adverse event reporting relies heavily on international non-proprietary names, or INNs. When an originator and biosimilars share the same INN, a reported adverse event without a brand name cannot be traced to the specific manufacturer. To separate this data, regulators enforce recording specific brand names, batch numbers, and unique identifiers, like the FDA's four-letter suffix. Global efforts exist to harmonize this data tracking, such as the World Health Organization's proposed biological qualifier system, though international adoption has been slow. This precise traceability is especially vital now, as healthcare systems increasingly mandate switching from originators to biosimilars at the pharmacy level. Without exact, standardized lot and batch tracking built directly into electronic health records, the adverse event data pool becomes polluted. This permanent masking hides the critical immunogenic safety signals we built the system to catch. Traditional passive spontaneous reporting suffers from systemic underreporting across all pharmaceuticals. For biosimilars, it is entirely inadequate. The data pool is further complicated by the nocebo effect during patient substitution. Patients who expect adverse effects due to a change in treatment often report subjective symptoms, which confuses the data pool. The modern solution is aggressively leveraging real-world evidence. Teams actively integrate vast data streams from electronic health records, insurance claims, and patient registries. By applying advanced artificial intelligence and data mining, pharmacovigilance teams continuously scan medical literature and global digital channels to bypass clinical underreporting. These proactive systems form an adaptive, living risk management plan. As real-world data accumulates, the surveillance protocols evolve continuously with the drug's life cycle. Biosimilars offer incredible economic savings and heavily expand patient access to life-saving therapies. A biosimilar's affordability is ultimately useless without the uncompromising traceability and specialized surveillance architecture required to guarantee its continuous safety to patients worldwide.